Hi kids, so now let us talk about your specific electrolytes since we are done with your introduction. So first two electrolytes will be your sodium and potassium. So this is considered as your lesson 6, the second topic for your midterm period. So as what I have mentioned in the previous video lecture, in terms of your specific electrolytes, we will be focusing on a brief description about each electrolyte, and then we will be focusing on the determination of your determination or method, and then the reference range, and then you have your specimen consideration or your sources of error. So starting now with sodium. Now, as you can see, sodium is a monovalent cation. Monovalent meaning they only have one valence, and that it is your most abundant cation in the ECF. Now, 90% of all ECF cations comprises your sodium, and because of this, it is considered as a major determinant of your plasma osmolality. Due to its high concentration in the ECF, the increase or decrease in the amount of your sodium directly affects the movement also of your water. So looking at your graph once again, kids, 'di ba? This is the your sodium na concentration in which, as you can see, whether it is in your blood plasma or your interstitial fluid, which is both comprises your extracellular fluid, so ingani gito siya kadaghan. Looking at your intracellular fluid, here, 'di ba, grabe lang kagamay ang amount of sodium that can be found in your intracellular. So even if you try to look at your chloride here in your bar graph, chloride is around 117 and 100, but then again, mas taas gihapon ang inyuhang sodium. So now let us proceed to the determination of your sodium. So we have your chemical methods, your FES, your EAS, and your ICE. So please do not forget the definition of these acronyms. For FES, that is your flame emission spectrophotometry. For your AAS, that is your atomic absorption spectrophotometry. And for your ICE, that is your ion selective electrode. Now, for your FES and AAS, these are two examples of your spectrophotometry in which, from the name itself, you can already see the difference of these two in terms of the principle and in terms of the light source. Diba? Flame emission, meaning flame, so its light source is a flame, while your atomic absorption, kids, its light source is your hollow cathode lamp. Now, another thing to remember about the difference between flame emission and your atomic absorption is the words are the words emission and absorption, kids. When we say emission, it is when your electrolytes return to an energy level. When we say absorption, it is when your electrolytes gain energy and jump to a higher energy level. So from there, di ba, makita na ang difference in principles on how they are going to measure your substances, your molecules, or your ions. Now, how about your ISE? ISE now, kids, is the most commonly used method in terms of determining not just your sodium but also your electrolytes in general. Chemical methods can still be used, but chemical methods are very outdated in a sense na we have a lot of disadvantages to look at when we perform chemical methods. And number one is in terms of the amount of volume needed in order to perform the test. And if you could still recall in terms of the discussion that we had on your Pentra C200, di ba na-mention na ako na your Pentra can also be used in order to measure your electrolytes, which is under the principle of your potentiometry, and that is with the use of your ion selective electrode. Now, the ion selective electrode commonly used for the determination of your sodium would be your glass ion exchange membrane. So let us begin with your chemical methods. So there are two chemical methods that can be used for sodium determination. You have your Albanese lane and you have your magnesium uranyl method. Now, these two, kids, are you just need to remember in terms of the reagents, that is needed in order to have a end color product for its measurement. And also take note that there is a protein precipitation that needs to be done in these tests. Just take note na number one interference sa mga chemical methods gyud ang inyong protein. And here in your electrolytes na mga determination, so it is common gyud na before reading your sample, there should be a protein precipitation that needs to be done. So, principle sa tauna, kids, let us start with your Albanese lane. So, for the principle, your sodium is made to react with your zinc uranyl acetate, in which a product will form, which is your sodium uranyl acetate precipitate, after the addition of your polyvinyl alcohol. And then with the addition of water, a yellow solution will form, and this yellow solution will then be measured using your spectrophotometer. So the protein precipitant being used here in your Albanese lane is what we call your TCA, or your trichloroacetic acid. Yun. So next we have your magnesium uranyl. So here sodium is precipitated with your magnesium uranyl acetate, and then the uranyl ions remaining in your suspension will form a yellow-brown complex upon the addition of your thioglycolic. Now the difference between your reagent blank and your analysis is proportional to your sodium concentration. So remember that there are a lot of types of blank, diba? As what I have mentioned, you have na-mention ako na siya during our discussion regarding your semi-automated machine, diba, naamoy water blank, naamoy air blank, naamoy reagent blank, and then naapod mo sample blank. So, depende na lang as to what is being required sa method na ato ang gina-talk about. So, here, in your magnesium uranyl, what is being used is your reagent blank. And then you also have your protein precipitant here. So our protein precipitant dalawa, you have your uranyl acetate and you have your magnesium acetate. So that's it for your chemical methods. So now we proceed with your ISE. Now your ion selective electrode kits are the following. So depende siya. If you could still recall your clinical chemistry 1, di ba natin mga ginatawag na dry chem ug wet chem? When we say wet chemistry, ang analyzing processes ani kids involved a liquid state, whereas when we say dry chemistry, instead of using a liquid, kumbaga, katong mga reagent na ginagamit nato is already being incorporated in a pad. So this one is an example of your wet analyzer or wet chemistry, and then this one is your dry chemistry. So as you can see here on your figure 16-2 na image on your left side, this is a diagram of your sodium ion selective electrode with a glass capillary membrane being used. While here on the other one, your figure 16-3, this is an example of a dry chem kasi this is a kahit na ion selective electrode pa rin ito kids, but this is a dry chem na tipong nakakaset tape siya na impregnated na dito sa mga testing pads nyo ito yung mga reagents na needed for us to be able to measure your sodium. So, yun lang naman. And in terms of your ISE kids, remember or recall the words kasi important din ito since we are talking about electrodes. It is important to remember that there are two types of electrode being incorporated. Especially if we are going to use ISEs in your potentiometry, you have your reference electrode and you have your measuring or your what we call your indicator electrode. So muna kung mapansin ninyo dirian ng mga images, diba na mo yung reference electrode na na-mention, even here na mo yung reference layer and then na mo kaning measuring or indicating electrode. Now, in terms of this electrodes, kids, what you need to remember is that your reference electrode, from the name itself, reference, so meaning siya ang parang standard or basis natin, and it is the electrode with a constant potential. Whereas your measuring or indicator electrode, so from the word itself na measuring, so siya yung nagre-respond sa mga changes sa activity dun sa ating solution. So, ayun yun. And also, you need to remember that there are also two types of ion selective electrode test. You have your direct measurement and indirect. Again, direct natay ginatawag na direct ISE and indirect ISEs. Now, the main difference lang mangyud between the two kids, no, when we say direct, meaning to say there is no need for you to dilute your sample, whereas when we say indirect, dilution is needed. So again, for direct, this is undiluted. For your indirect, ito needed na diluted. And between the two, what is commonly used is your direct ISE. So actually, there is not much difference in terms of the results as stated by Bishop. Now, there is no significant difference daw in the results, except lang yun when your samples are hyperlipidemic or hyperproteinemic. Again, if your except lang if your samples are hyperlipidemic or hyperproteinemic, so in that case, uh, better use your direct. Sige lang, let me just, kids ha, double check that one. But as far as I could recall, what Bishop mentioned is better use your direct ISE in that cases. And just remember also, kasi I think na-discuss naman din talaga ito, kasi nga mga principles na mga nyo to, mga principles of instrumentation, that one source of error with your ISAs is really in terms of protein buildup. So, kaya every now and then, dapat may maintenance din na ginagamit dun sa mga electrodes nyo. So, again, one source of error with your ISE is protein buildup. Especially if it is being used continuously. So, ayun yun siya. So now let us proceed with sample considerations. So what are the things that you need to remember before measuring your sodium? Serum, plasma, and urine are all acceptable for your sodium measurement. Hemolysis does not cause a significant change in serum or plasma values as a result of decreased levels of intracellular sodium. Only if there is a marked hemolysis na we need to do repeat collection of your patient. If there is marked hemolysis, levels may be decreased as a result of a dilutional effect. So actually sa ako ang ihatag na laboratory output sa inyo ha kids na ipa-photocopy, there is also a portion there in the introduction part sa inyong lab output wherein naatay mga sample considerations pod nag-emphasize. So, na-mention man nato ni si number 1. Number 2, so when plasma is used, lithium heparin, ammonium heparin, and lithium oxalates are suitable anticoagulants. And then for number 3, timed collections of urine, feces, or gastrointestinal fluids are preferred to allow comparison of values with reference intervals and to determine rates of electrolyte loss. And actually, kids, this is not just for sodium. In terms of trying to measure analytes mga analytes in the urine, it will always be best to have a 24-hour urine collection para better ang estimation sa analyte. Kesa dun tayo sa random na urine lang or isa lang dayon ka sample na katong first lang ninyo na micturation. So yun siya. And then number 4, serum plasma and urine may be stored at 2 to 4 degrees Celsius or frozen. Number 5, erythrocytes contain only 1/10 of your sodium. So ito po yung reason as to why hemolysis does not cause any significant errors in your serum. Because remember, asa makita inyong sodium nasa extracellular. So that is why dili kaayo ing anak ka dako ang problem if you have a hemolyzed sample tapos mag-sodium determination mo. Lipemic sample, lastly, lipemic sample should be ultracentrifuged and the supernatant analyzed unless a direct ISA is being used. So I think this already answers yung ating concern kanina, di ba? Na walang difference between direct and indirect except lang pag hyperlipidemic or hyperproteinemic. And as stated here, if lipemic yung sample, better use direct. So nasagot na natin yung concern kanina. Sige, so for the reference range, for serum or plasma, 136 to 145 millimoles per liter. If it is a 24-hour urine sample, so you have 40 to 220 millimoles per day. That mo na siya meaning ha sa letter D. which may also vary depende sa diet. And then for your CSF, you have 136 to 150 millimoles per liter. So, syempre, you need to look at your package inserts. Although, I guess the problem here sa atua, karon sa clinical chemistry lab is that there is really no specific reagent mentioned in terms of what is being used in sodium. So that is why let us stick na lang with your humalizer na brand. And as you can see here, kids, this is of course a chemical method. And what is used here in the chemical method of your sodium is yung second, yung magnesium uranyl nyo. So, ayun. No? Photometric determination of serum sodium, magnesium uranyl acetate method, and of course, it is a colorimetric test. So you have here your method. Sodium is precipitated with your magnesium uranyl acetate. Your uranyl ions remaining in the suspension will form a yellow-brown complex with thioglycolic acid. So yan po yung na-mention natin kanina in terms of our magnesium uranyl na method. And then, ito po yung mga reagents nyo. So, your precipitating reagent, di ba? Your protein precipitant is your uranyl acetate and your magnesium acetate. Then, the purpose of adding thioglycolic acid, not thioglycolic, the purpose of adding your ammonium thioglycolate as seen here is that it will be the substance that will impart a color in your solution, and thus your ammonium thioglycolate magiging thioglycolic acid as mentioned in the principle. Then, syempre, you have your standard. So, in terms of stability, stored and opened at room temp and in the dark, the contents are usable until the expiry date. And then, so yun, na-emphasize pa rin po dito na as much as possible, your reagent is nasa dark or walang light. And then, specimen here, kids, ito. So, if ever man na mag-sodium determination ta na chemical method, so dapat serum. Kasi as indicated here, serum lang din po talaga yung nakalagay. And dito, kids, so for your wavelength, best to use is your 410 nm. Kasi nga, looking at your 365 and 405, di ba naka-mercuric nanometer siya. So, just to be sure, pwede ta mag 410 nm na lang. And then, sa pa ba? Ayun, in terms of the measurement, it is measured against your reagent blank. And this is now your pipetting scheme. So, as you can see here in the pipetting scheme, kids, medyo trabahuso siya in a sense that kailangan manggod ta mag-precipitate at the beginning. So, as you can see here, di ba, naatay prec. So, take note na here, prec stands for your precipitating solution. So, muna na pinakauna na dapat buhaton is to do precipitation, after which, sa kapata, mag-add kato sa inyuhang color reagent. So, here, macro and semi-micro kids here are referring to the volumes lang, and always it would be best to use semi-micro para dili pud ingana ka daghan ang inyuhang amount of precipitating solution, and even your sample na volume gagmay lang yun siya. So diba, as you can see the difference. For your serum sample sa semi-micro 20 lang, compare it with the 50 that is needed for macro. And then for your precipitating solution, 1,000 for semi-micro, 3,000 for your macro. So better use those with smaller volume. So here, as you can see sa procedure, sorry kids ha kung dili ko makagamit sa ako ang pen kay dili sa bayan di ay ang paggamit sa pag-zoom in ug ang kanang sa pen here in your PowerPoint. So I hope you can still follow. So as you can see here, close tubes and mix well. After adding your sample and your precipitating solution, you need to close your tube and mix it well, then allow to stand it for 5 minutes. Now shake intensively for at least 30 seconds, and then allow to stand for 30 minutes. Now centrifuge at high speed for 5 to 10. Now after ma-centrifuge, kids, yung clear na supernatant yung gagamitin natin. So here, clear supernatant around 20 microliter, and then add 1,000 microliter of your color reagent. So mix well, and after 5 to 30 minutes, measure absorbance of your reagent blank. then your standard, then your sample against distilled water at 360 to 410. So, dito tayo sa calculation nyo. So, in order to get the result of your sodium, so makita siya kids, very important na may absorbance po tayo sa reagent blank. Kasi yung absorbance ng reagent blank natin i-minus sa sample and sa standard. So, kaya important to get all the values of the three for us to be able to compute and get the concentration of your sodium. So, ang ating multiplication factor here, kids, is 150 for millimoles per liter. So, syempre, in terms of linearity, sodium concentration exceeding 300 millimoles per liter must be pre-diluted 1:1 with distilled water, then multiply the results by 2. So, depende sa ating dilution na ginawa kung ano yung ating dilution factor. And then your normal range is 135 to 155 millimoles per liter. So, naalam po tayong mga notes diri. Use the semi-micro procedure only if a very efficient centrifuge that can that has a ingani ang speed, iyang rotations per minute from 8,000 to 10,000 is available. Di ba? Kaya man na sa centrifuge na ginagamit nato sa lab. And then otherwise, use the macro procedure to obtain reliable results. Your prec becomes discolored when exposed to light. So, store protected from light. Its light turbidity does not affect the determination. So, ito lang din kids ha. Kasi baka magtaka kayo, now the reason why it was emphasized to really put your reagents in a dark place, it's because of your precipitating solution as discussed here in the notes. And then detergents usually contain high sodium concentration, so the equipment, your test tube, your pipettes, all the equipments being used in order to measure your sodium must therefore be rinsed carefully with distilled water and then avoid contamination by traces of sodium, especially your sweat. Kasi syempre, di ba, yung sweat natin that is made up of sodium chloride, so may sodium pa rin, so that could affect your test result. So disposable plastic tubes are recommended for the determination. Use parafilm or plastic stoppers to close your tube. So common naman din po talaga yung parafilm sa atin. And then, then you have your references. So that's it for your sodium. So now let's proceed with your potassium. Now, potassium kids, if major extracellular cation ng si sodium, potassium is the opposite. Potassium is your major intracellular cation. And being an intracellular na cation, meaning to say it is being housed within your cells. So, siya yung nasa loob ng cells natin. And the concentration of your potassium is 20 times greater inside than in the outside of your cell. So, again, yun na nga, intracellular siya. So, only 2% of total body potassium circulates in our plasma. Now, try to look at your bar graph. Diba, saan po yung potassium? Andito. Try to compare it with your intracellular concentration of sodium, tapos kani kay potassium. So, grabe yun kataas ang concentration ni potassium inside your cell. So, thus, it is called as your major intracellular cation. And diba, our kids, less than 1/4 pamanggani ang volume ni potassium outside or sa extracellular fluid ninyo. So they are, sodium and potassium are the total opposites with each other. So with potassium kids, let us begin first discussing the sample considerations. So here, for the collection of specimens and even in specimen handling, daghan ta og i-consider kay potassium kits. Number 1, in terms of the coagulation. Now, coagulation processes releases your potassium from your platelets. Kasi syempre, platelet is a type of cell. So, meaning to say, na potassium na daghan sa iyahang sulod. Once your coagulation is activated, once your potassium is activated, ma-release man gud na ang inyuhang potassium. So as you can see, serum potassium may be 0.1 to 0.5 millimoles per liter higher than your plasma potassium concentration. So kaya mas maganda sana if plasma yung ating sample for potassium. But then again, that's a case-to-case basis. Depende siya sa protocol sa hospital. Depende din po siya sa ginagamit natin sa lab na machines. Kasi binabagay-bagay din siya. And the next, thrombocytosis will further elevate your potassium. And actually, this statement, kids, is the same with this third one. Medyo na ano lang siguro ako during paggawa or pag-type nito. But then again, the second and the third one are actually the same because when we say thrombocytosis, that means that there is an increase in your platelet count. Then again, if there is an increase in your platelet count, there is also an increase in your potassium levels. So here, better use if in cases na si patient may thrombocytosis, better use heparin as your anticoagulant of choice. Common good po si heparin, kids, sa treatment ani ng mga cases na naay mga coagulation problems. So that is why in terms of measurement, mas pinipili na rin si heparin. So next, prolonged tourniquet application or fist clenching could actually increase your potassium as there will be a shift of the volume of your substances. And then if there is hemolysis, take note, hemolysis would mean that there is a destruction of your red blood cells, thus releasing your potassium, thus further increasing the amount of your potassium. And actually, kids, gitry gyud na ako ni siya kay due to my curiosity regarding the difference in the levels of your potassium, if normal or hemolyzed ang sample. Nga dito one time na katong nag-work pa ko sa laboratory and then naman gud mi hemolyzed na sample, gitry gyud na ako siya og run. Same patient, normal og kanang hemolyzed na sample and grabe gyud ang difference. So that is why really prevent using hemolyzed na sample if you are trying to check for the concentration of your potassium. Next, whole blood samples should be stored at room temp and analyzed as soon as possible. Why? If you will place an ice dito sa inyuhang sample na need for potassium determination, it will promote the release of your potassium, thus falsely elevating its concentration. So still in your laboratory output, kids, that I will give next meeting, may portion pa rin po kayo dun sa sample consideration. So kindly read that one. And actually, most of this na-mention na kanina, but just to emphasize further, so number one, hemolysis of 0.5% of RBC can already increase the level of your potassium by 0.5 or around 30% increase. Potassium concentrations in plasma and whole blood are 0.1 to 0.7 millimoles per liter lower than those in serum, and stated reference intervals for serum potassium are 0.2 to 0.5 millimoles per liter higher than those your plasma potassium. So plasma levels are lower compared to serum levels because of the release of platelets into serum on clot formation. So na-explain na ako ni siya kaganina. So yun na nga, case-to-case basis as to the protocol of hospitals. Basta dili lang yun mo mag-worry, kids, kay naagyo designated na reference range kung serum or kung plasma ang ginagamit. Next, 10 to 20% increase as a result of muscle activity. If there is a mild to moderate exercise, ani ang level na itaas sa inyong potassium. If there is vigorous exercise or usually kaning inyuhang fist clenching, there is around 2 to 3 na millimoles per liter na increase. So basically, the reason behind this, kids, is that your potassium is actually involved in the sending of electrical signals in your muscles. So it lets them contract your muscles properly. So if low ka og potassium levels, then you would expect that there is weakness and cramping or kanang mga muscle cramps na mahitabo sa imuhang body. Number 4, prolonged contact of bawal prolonged contact of serum and red cell, bawal prolonged application of tourniquet. Again, both of this will cause an false increase in your potassium. Now, number 6, if a whole blood specimen is chilled before separation, glycolysis is inhibited, and the energy-dependent sodium-potassium ATPase cannot maintain this gradient, and increases in plasma potassium will occur as a result of potassium leakage from your erythrocytes and other cells. So ito na po yung explanation kanina sa na-mention ko po regarding why bawal i-ice or bawal i-chill yung sample nyo for potassium measurement. So the increase of potassium in serum is of the order of 0.2 millimoles in 1.5 hours at 25 degrees Celsius, whereas at 4 degrees Celsius, the increase is considerably greater and has been reported to be as much as a 2 millimoles per liter increase after 4 hours at 4 degrees Celsius. So consider that one. Dako na kayo ng maskin isa lang kids, maski 1 point na increase ng dako na kayo na siya na difference, especially if we are talking about accuracy. Now, lastly, a falsely decreased potassium value is initially observed if an unseparated sample is stored at 37 degrees Celsius because glycolysis occurs and potassium shifts intracellularly. So that is why it is always encouraged to use a uh, serum separator na mga tubes kanang naay mga separator, mga gel separators ninyo kids, and di ba nakakita nakagamit naman gyud mo ana. So dako jud kaayo ang tabang ana niya, not just to prevent na mag-mix balik, ma-disturb and ma-mix balik ang inyong gicentrifuge sa sample, but also for this reason. So, for the determination of your potassium, ito lang naman din kids yung tatandaan nyo. Meron din tayong chemical or colorimetric method in which we call that one your Lockheed and Purcell method. So here, the reagent used is your sodium cobaltinitrite, which will now form your sodium potassium cobaltinitrite. Now, with the addition of your phenol, which is considered your color developer, a blue color will be observed, and the production of this blue color will be measured using your spectrophotometer. We also have ISA testing here, your ion selective electrode. So kung glass ang sa inyuhang sodium, for your potassium, what we use is your valinomycin na membrane. And we also have your potassium chloride as your inner electrolyte na solution. So for the reference range, for serum that is 3.5 to 5.1, for plasma, depende kung babae or lalaki. So males, 3.5 to 4.5, females, 3.4 to 4.4. So actually, gamay raman og difference, no? And then for the 24-hour urine, you have your 25 to 125 millimoles per day. So dirita sa package insert sa inyuhang potassium. So as you can see here, what is being used for the measurement is a turbidimetric na method, pero actually somehow related gamay sa katong inyuhang colorimetric method kanina na sa Lockheed and Purcell nyo, but try to look at the reagents being used for your potassium determination. So still, we will be doing precipitation here, protein precipitation here for your potassium determination. So the precipitant here for potassium is your TCA, your trichloroacetic acid. Then your reagents would be your sodium tetraphenyl boron and your sodium hydroxide. And then, of course, you will always have a standard to be used. So, first thing, before performing the mixing of your sample and reagent, since we have two reagents presented here, we need to do a reagent preparation first. So, you need to mix the content of your bottles dun sa TPB nyo, your tetraphenyl boron, with the content of your sodium hydroxide. And then, na-emphasize dito po yung ratio. So, 1:1 lang. So, kung mag 1 ml mo sa inyuhang tetraphenyl boron, then 1 ml po sa inyuhang sodium hydroxide na reagent. And then, before using that mixed reagent, kids, make sure na i-allow sa ninyo siya to be completely mixed. Allow to stand for 15 to 30 minutes. Saka pa nato siya exactly na magamit. So, diri na dayon ta sa assay ninyo. So, as you can see, your wavelength is 578. And then, still in terms of its measurement, against reagent blank gihapon siya. So, kindly look at your pipetting scheme. So, as you can see here, precipitation sa gyud ta una, 50 microliter for your sample, and then 500 microliter for your precipitating agent. Mix carefully, centrifuge at high speed for 5 to 10 minutes. And then after, yung supernatant yung gagamitin natin, mix with the working reagent or yung reagent na ating prepare. And then 100 microliter for the supernatant, 1000 microliter for the working reagent. So here in the procedure below, to produce a homogeneous turbidity, standard or the clear supernatant have to be added to the center of the surface of your working reagent in the cuvette. So mix each cuvette carefully before proceeding to the next sample and allow to stand for at least 5 minutes before we could actually begin its determination or bago natin ilagay dun sa spektro nyo. So dito, uh, yun. So here, di na kailangan isubtract yung reagent blank nyo. So bali, as seen here in your calculation, all you need is your sample and standard na values sa absorbance, and then either millimoles per liter or milliequivalents per liter, kids, same multiplication factor, that is 5. So na-emphasize din po dito yung normal values nyo. For serum, that is 3.6 to 5.5. For plasma, that is 4 to 4.8. And the same ang gihapon in terms of linearity, kids. Again, even di ba sa inyuhang machine na amang gani siya limit up to asa lang ang iyahang kaya ma-read na absorbance. So the same way with your chemical methods, if ever na dili man ma-read, so that would only means na taas na kaayo ang inyuhang absorbance na result, so a dilution is needed. And then, saba, for the notes, use non-hemolytic serum or heparin plasma as a specimen. As red blood cells contain about 25 times the amount of potassium, they have to be separated from your serum within an hour after blood collection. Otherwise, falsely elevated potassium concentrations will be found. And then traces of detergents produce turbidity. Syempre, we all know that. So which leads to falsely elevated potassium concentration, so therefore should be avoided. Contaminated glassware is the greatest source of error, kids. Glassware should therefore be thoroughly rinsed with the ionized water, actually kahit distilled water na. Disposable plastics ware may contain softeners which react with the reagent and is therefore not recommended. So ana ka kuti ang inyuhang electrolyte na measurement, especially if we are doing manual gyud na kanang processing, kids, labi na sa kaning in terms of mixing. So that's it for your sodium and potassium, kids. So for the discussion regarding your fully automated na machine, so we'll just create a different na pod na video lecture kay para at least na pod mo short na break. So thank you for listening, and then next na pod ta na video lecture.